Okay, this is Lab 7, Digestive System Part 1. We do this in two parts. The first part is a significant amount of structure function, starting at the mouth and kind of working our way through the entire alimentary canal to identify what you need to identify and basic functions. When it comes to digestion 2, we're going to talk about the chemical and enzymatic digestion of food. And again, looking at some of these same organs and see what their contribution is to digestion. Okay, so we are going to start with digestive anatomy, and we're going to identify all these bold printed items: the oral cavity, tongue, teeth, salivary glands, parotid glands, subungular glands, all of these. And of course, there is functions to match the physiology. So this is going to be easy enough to be able to identify multiple models as we progress through. We can start looking at the images. So let's start on the functions. Okay, so as usual, we start with the options of the descriptions, and it's easier to then match it to the particular structure. A, produces amylase and moistens food for swallowing. So if we're moistening food for swallowing, we must be in the oral cavity. And so that would be salivary glands. So A goes with salivary glands. We do have three different ones we'll look at: parotid, sublinguals, and submandibular, but as a family, salivary glands. B, increases the volume of the stomach. That's referring to the rugae. The rugae are folds of stomach tissue that you see that are evident when the stomach is pretty much empty of any food. It's like a collapsed balloon. Then when you eat food, it stretches the stomach, kind of like blowing up a balloon, where the skin of the balloon becomes more translucent. As the stomach expands, those rugae disappear, and now it becomes kind of a smoother surface. And so the rugae are the folds we're going to see into the inside mucosa of the stomach. C, forms feces, absorbs water, vitamins, and electrolytes. That is the large intestine. D, stores and concentrates bile. That would be the gallbladder. E, mastication of food. That would be teeth up at the top. Mastication is just a fancy word for chewing. F, completes digestion and most absorption of nutrients. That's the small intestine. G, where peristalsis begins and transports food to the stomach. That's the esophagus. H, interconverts nutrients, produces bile, detoxifies substances like drugs, alcohol, produces plasma proteins, produces anti-A's and anti-B antibodies. When you see a fairly lengthy list of functions, it's the liver, because the liver has about 503 different functions. So H is the liver. I, regulates entry of chyme into the duodenum. Well, first of all, chyme is the watery solution in the stomach after it's been churning food that you've eaten. And so we call it chyme. And that passes from the stomach into the duodenum via the pyloric sphincter. Pyloric sphincter. J, common pathway for food and air. That would be the pharynx. Like we said, the throat area. We have, where food and air passes. That's the oropharynx and the laryngopharynx. So I believe that's up here on the top part. K, prevents chyme from entering the esophagus. That would be the cardiac sphincter. term we used here, esophageal or cardiac sphincter. L allows materials from the small intestine to enter the large intestine. That's the ileocecal valve. M manipulates food for swallowing, contains taste buds, forms or helps you articulate words, not words, words. And that would be the tongue. And increase in surface area in the small intestine. Right. So the small intestine has modifications there as well. That would be villi. O, storage site for food and where protein digestion begins. That is the stomach. P produces enzymes. Bicarbonate produces hormones to regulate carbohydrate metabolism. So which one produces the bicarbonate and which ones produce, and really we should be a little more specific about that, produces insulin and glucagon. That would be ding, ding, ding. That's your pancreas. And Q transports bile and pancreatic juices into the small intestine. That would be the pancreatic duct. Now, we used to have an option just for use hepatopancreatic ampulla or sphincter. That's the point that it enters. We'll look at a picture of that. The ampulla is a region, it's sort of like this, along the pathway of the lymphatic system. We looked at the chyli in the abdomen area, the cisterna chyli, that is a dilated region in the lumbar area where multiple lymphatic vessels merge, and it has to dilate before it narrows down, and from there superiorly becomes the thoracic duct. Well, where the common bile duct merges with the pancreatic duct, the same phenomena kind of happens there. So as we're transporting all this bile from the liver and or concentrated from the gallbladder, it's going to pass through the common bile duct, and that merges with the pancreatic duct. And when it does, again, it kind of needs a dilated region to accommodate the excess volume, of fluids traveling along the bile system as well as the pancreatic duct system. So it dilates, and that's called the hepatopancreatic ampulla. As it passes and merges, makes contact, so to speak, or intersects the small intestine, there's another little sphincter of muscle there, and that is the hepatopancreatic sphincter. So that can close down or relax and regulate the amount of bile and pancreatic juices that are entering the small intestine. Okay. All right. When it comes to digestive system histology, the only one I ask you about is the small intestine, because the stomach and the small intestine really look very, very similar. More critical, I find the functions occur in the small intestine, and it's a longer system and tube. So we're going to look at the histology of the small intestine. We're going to look at the innermost layer that's, sort of intimate with the lumen of the tube, and that would be the mucosa layer. Just like in the trachea, you have a mucosa, submucosa. Well, we have a mucosa that's composed of simple columnar epithelium and are found on villi, which are finger-like extensions of mucosa, that extend into the lumen. And picture, instead of the small intestine having a nice flat surface, like looking in inside a pipe, let's say a firm pipe, picture looking down this pipe and then having hairbrush bristles extending into the lumen, right, from the entire circumference of that structure. And that's what the villi kind of look like. They extend inward, they help increase the absorption that can occur there, or your absorption area. It also helps slow down the transit of chyme through the small intestine so that enzymes that are being liberated onto them can have time to enzymatically do what they need to do chemically, and produce certain products that can be absorbed in the small intestine. Okay, then we have the submucosal layer that is composed of areolar connective tissue and simple cuboidal epithelial tissue, because we will have some mucus glands there. So far, very similar to the trachea. Then we have a muscularis layer. Now, the muscularis layer is going to have two layers of muscle. We have this inner circular muscle layer and the outer longitudinal muscle layer. So the first layer of muscle, that would be kind of intimate with the submucosal layer and mostly areolar connective tissue there, is the circular. Superficial to that, so to speak, is our longitudinal muscle. And when we look at it on the slides and on the models, it shows the tissue, these muscle cells kind of coursing in different directions, so you can see the difference between one muscle layer to another. And then the most outer layer is the serosa, and that's composed of areolar connective tissue. Once we're in the abdominal pelvic cavity, or we're pretty much outside of the thoracic cavity, within the thoracic cavity, we call it an adventitia. Once you're outside of the thoracic cavity, you call it a serosa. And that outer serosal layer is composed of areolar connective tissue. All right, then we're going to look at the pancreas again, looking at the acinar cells of the acini cells and the pancreatic duct. Remember, we looked at it the previous lab because we're looking at the islets of Langerhans, where alpha and beta cells were, and I mentioned that those other darker purple, cells there were acinar cells, and they function in the digestive system, and thus the pancreas is like a dual system functioning organ. It has components of it that function in the endocrine system and some that function in the digestive system. Right, then we'll look at a liver. We're going to look at the lobule, which is the repeating unit of the liver. Kind of looks like a stop sign. Remember, in compact bone, we saw osteons that repeated over and over again. Now we're going to see a lobule. Kind of has a hexagonal shape. And within it, you're to identify hepatocytes, which are liver cells, the central vein, and the sinusoids. The sinusoids are a type of, I guess you could call capillary, except for capillaries, because blood is going to pass through this kind of a tube system, but it's not composed of simple squamous epithelium. It's composed of those hepatocytes. So the hepatocytes kind of form the walls of these like liver capillaries and allows blood to pass through. Where multiple lobules, or where lobules meet and form kind of a wall from one lobule to another, these junctions, we have hepatic triads, and that is composed of these three structures: the bile duct, hepatic portovein, and the hepatic artery. When we were looking at blood vessels in exam 1 material, we said if you looked on the undersurface of the liver by the gallbladder, you'll see entering and somewhat exiting the liver, coupled together. You're going to have one green vessel, a blue vessel, and a red vessel. Hepatic triad. Outside the liver, right, that's going to be a bile duct or a hepatic duct, and hepatic artery and the hepatic portal vein. Once you're in the liver, we just call it a bile duct. Location is going to determine what you call that duct. Within the liver itself is the bile duct. Outside of the liver, we're going to look at what's called the biliary tree, the ducts that are transporting bile away from the liver, because the material in the green tube system is being transported away from the liver, not to the liver, like the blood in the hepatic portal vein and the hepatic artery are. So we have a right and left hepatic duct that form a common hepatic duct. And then that merges with the cystic duct that's transporting bile to and from the gallbladder. And those two cystic and common hepatic ducts merge to form a common bile duct, which then travels into the pancreas and merges with the pancreatic duct. I know it's a lot of little precision, but it's like naming the arteries that, depending on the region that you're in, is going to determine the name that's assigned to that structure. Okay, then we're going to look briefly at teeth. Primarily what you need to know is to be able to identify the crown versus the root area. So here, over to the right-hand side, above the gum line is the crown, below the gum line would be the roots. And you can see, these roots here. The enamel is the outer white portion, generally. It's a very, very hard substance. And within it, we have pulp, we have nerves and blood vessels, as I'm sure most of you know, and we have ligaments that hold them in place called periodontal ligaments. Okay, so we've now looked at the crown and the root. The crown, I don't think, I wouldn't really worry too much about the function, but the crown, is the region, above the gum line that contains enamel. It's the hardest part of the tooth. And then the root are the extensions below the gum line that contain nerves and arteries and periodontal ligaments that keep the tooth in that tooth socket, or in the alveolar process, it's called. Okay, then we have our teeth. We have incisors, canines, premolars, and molars, or cuspids and bicuspids. So the incisors, the ones to the front that are flat like a knife, they cut. Canines are the sharper teeth. Some people refer to them as eye teeth, but they're the sharper fang-like ones, right? To the two on the upper right and left, or I should say two on the upper row of teeth, one on the right, one on the left, two on the lower row of teeth, one to the right, one to the left. Those are shredders. They shred. And then the premolars and the molars, we generally have on one upper right-hand side, which would be identical to upper left, and the lower right and the lower left. You had two premolars and then three molars to each quadrant: upper right, upper left, lower left. Premolars and molars, they're grinders, right? They're grinders. They macerate between them by, moving the teeth back and forth. Right, so we have incisors that cut, canines that shred, and premolars and molars that grind. Kind of like a mortar and pestle. You know what that is? Okay, so that is let me stop this share. So that's our exercises all filled out. Let's move to all right, let's move to images. There we go. Just let that kick in. So there we go. All right. We're starting off with another big cookie sheet model, one of those big plastic ones. Once again, share. There we go. Okay, so picture this right side up. And here we have the mouth, the whole digestive system, looking at the head there where food enters the mouth, and we can see some of the salivary glands there. Then that leads to the esophagus, which is the muscular tube you're looking at here, to the stomach, and we can follow the entire digestive system to the stomach, the liver to the upper right-hand quadrant, and the gallbladder beneath it in green. We have the small intestine, regions of the large intestine, and then the rectum. To the upper left-hand side, we see a very large stomach there, shows you the 3 layers of muscle found there. I would know those 3 layers. We see the entrance point and the exit point. We'll see more pictures of these. So we have the stomach, and you can see the green gallbladder over to the side. We can see part of the duct system, the cystic duct, the common hepatic duct, and the common bile duct. And then on the bottom is showing you the villi, the large villi. And here's another model. These are really good models. We have another one that's the newer version of this, and it identifies the same things, mostly just a little bit different, starting at the food being taken into the mouth and the oral cavity, the different regions of the oropharynx to laryngopharynx to the esophagus, stomach, liver, gallbladder inferior to it. We have the small intestine, large intestine, the pancreas, and ending with the rectum. When we're going to look at some of these functions, but most of these we will be looking at when we do perform the lab next week. But in some of these, there's a chart that will help you identify which organs produce which digestive enzymes. Digestive aids. So it may be identified as just DA as a digestive aid for a function. Just use E as enzyme and H as hormone. You'll see where that comes in. Okay, so here's one of those half heads. We generally looked at the opposite side when we were looking at the oral cavity, the nasopharynx, oropharynx, the laryngopharynx. Now we're looking at the lateral aspect and identifying the parotid gland. So that large gland that's superficial to your temporomandibular joint, that is your parotid gland. It's one of your salivary glands. It produces saliva and salivary amylase. So saliva acts as a digestive aid, because it moistens food, helps you swallow. And within saliva, we have the enzyme salivary amylase. And salivary amylase begins carbohydrate digestion. But we'll get into more of that, later. But while we're on the looking at these glands, it's just a good idea to identify, hey, functionally, this is what it does. The saliva, has multiple functions, but primarily is to moisten food. Most of it is water, so it allows food to be dissolved in an aqueous solution so that your chemoreceptors of your taste buds can respond to them, or detect their presence. And then we have salivary amylase is the digestive enzyme, for starting to break down a polysaccharide to a disaccharide. We'll look at that more, next week, or the next lab, I should say, not necessarily next week. This is another model that identifies, here's the tongue on the top. So there's a gland just below the tongue, and then there's another gland farther down that is below the mandible. So one is the sublingual, and the other is a submandibular gland, right? They produce saliva, as well, and salivary amylase. There's another model I might have in my PowerPoints that really I think is better color coded, to see all 3 at once. So we'll kind of zip through the notes toward the end. Right here, we're looking at the tongue. So you should identify the tongue, or identify number 1, it's the tongue. Then identify its function. It moves food around within the oral cavity so that we can push food between the teeth so we can chew food. It helps you initiate swallowing, so it's going to elevate and kind of push food toward the back of your throat. But remember, we also find taste buds there. Here we're looking at teeth. We have incisors, canines, and premolars. Incisor, canine, premolar. Come on. Here we go. Got a little stuck there for a moment. This is a really good model. So we have one is incisor. Two, now that we're on the lower, you can see it's a little more pointed. That's a canine. Premolars and then molars. So I would know, identify that tooth, it's an incisor. Identify the function of that tooth, cut. One is pointing to the esophagus, transports food to the stomach. The, the mechanism of propelling food down the esophagus is called peristalsis. Peristalsis is the coordinated contraction of muscle along this entire tube system, where the circular muscle contracts behind some food you're trying to swallow. Right? So you start pushing it down to the stomach, and distal to that quantity of food, the longitudinal muscle contracts and pops open the tube, so that when the circular muscle contracts behind it, there's no resistance, ahead of the food to keep it from moving down the tube. It's kind of hard to describe, but if you pop open the hose and you push behind it, the food is going to move into that region, with less effort. And then both of the muscle layers will contract, and you'll do that again. Longitudinal muscle distal to the bolus, the quantity of food you're swallowing, will contract and pop open the tube. And now the circular muscle behind it squeezes and pushes that little piece of food forward. All right, here we're looking at mostly the stomach. Right. And you have a lot of different numbers here, so that even though we don't have arrows necessarily pointing to things, it's an opportunity to look at a lot of anatomy here, while we have it nicely kind of a large picture to see them all. Well, first of all, you have 1, 2, and 3. So 1 and 3 are the entrance and exit points to the stomach. Right. So 1 is pointing to the lower esophageal or esophageal cardiac sphincter, and 3 is pointing to the pyloric sphincter. 2 is pointing to those humps and bumps in the mucosa, the inner layer of the stomach, and those are the rugae. However, this gives us an opportunity to point to 7, right, which is technically above number 1, above the sphincter, and that's referring to the esophagus that's connecting to that point. We see 3 layers of muscles to the stomach, 28, 27, and kind of 29 here. All right. The most inner layer is your oblique layer. 27 points to the circular layer, and 29 is pointing to the longitudinal layer. So there are 3 layers of muscle to the stomach, and that's because it needs to twist and turn and churn food around, like a washing machine, to increase the surface area, allow hydrochloric acid to mix with the food that you've eaten, let it mix with some mucus, facilitate absorption that will occur in the stomach. So we have 3 layers of muscle there. And then while we're here, 11, which is, just past arrow number 3, we have what's called the duodenum portion of the small intestine. And then behind it, you see all the green structures there. 10 is the gallbladder. 32 is the cystic duct that transports bile from the gallbladder to where 33 and 32 merge at 34. So 32 is the cystic duct, 33 is the common hepatic duct, and where they merge, because they're all heading now down inferiorly, behind that pyloric sphincter to the pancreas, right? Now we have the common bile duct. All right, so we have the cardiac sphincter or lower esophageal sphincter, or esophageal sphincter is fine. Technically, it's the lower esophageal. We have the region of the body of the stomach, but really what we're trying to point to is the rugae. And then we have the pyloric sphincter. The stomach as an organ, right, releases hydrochloric acid. It acts as a digestive aid. And hydrochloric acid has kind of multiple functions, but it starts to denature proteins. You know that if you decrease the pH of a solution, you start to break the hydrogen bonds. And when you eat these proteins that are very large, complicated chains of proteins, all amassed together. Remember, proteins are the organic family that demonstrates a primary, secondary, tertiary, and quaternary structure. So to start to break those hydrogen bonds and let these proteins start to kind of unravel, hydrochloric acid is really good at that. But it also helps, convert, another enzyme in the stomach from an inactive form to an active form. And we'll look at that more next week. It also helps to inhibit microbial growth, because bacteria and some bacteria and viruses are particularly susceptible to to low pH. So hydrochloric acid can help act as an antimicrobial. Pepsinogen is found in there. Right? That is an inactive form of pepsin. It's going to be converted by hydrochloric acid. And then the stomach also releases the hormone gastrin. But those we will primarily look at next week, because there'll be a chart that you're going to fill out using that information. Okay, here we're looking at a pancreas. Here's our pancreas. Over to the right, we have a spleen. So within the pancreas, we can see that white duct system. That's the white pancreatic duct, or pancreatic duct, but it's the white duct system you're looking at here. And it ultimately merges to the left with the green common hepatic duct, and will enter into the small intestine or the duodenum, the specific region of the duodenum of the small intestine. A down below is just pointing to another model type. In the labs, there's frequently different examples of organs from different models that we've had over the years. So in that particular model, the pancreas is white and the duct system is kind of that light bluish, light grayish kind of color to it. All right, so the pancreas, multiple functions. Again, this will be used in more detail next week. Again, we're going to put these in charts. The pancreas produces bicarbonate. The liver produces bile for emulsification of fats, and the pancreas produces bicarbonate, which helps neutralize that chyme, right? The fluid and the material passing from the stomach into the small intestine. Very, very acidic. But the pancreas produces multiple other enzymes associated with digestion. Produces pancreatic amylase, trypsin, chymotrypsin, carboxypeptidase, lipase, DNase, and RNase. What you should know right now is bicarbonate and some digestive enzymes. That's good. All right, here we're looking at where these structures meet, and even there's another model, one of that cookie sheet model that I keep referring to, that big plastic one, which was the first image we saw. We can get zoomed in on that. that gives you a really good view of how these two systems merge. So one is the pancreatic duct. It merges with the green there behind number 5. That's your common bile duct. And then it passes into the small intestine at the pointer 2, or arrow 2 is pointing to, I think that's a 6 right there. That little round structure, like with the red interior, that is the hepatopancreatic sphincter. I was just going to say is that just a little to the right of where that number 6 is, technically, that is where the ampulla would be found. So, depending on what your options are to answer, if it says hepatopancreatic sphincter / ampulla, then that would be fine. Okay, so here we have the pancreatic duct, hepatopancreatic sphincter, or the sphincter of Oddi. I still often refer to it as the sphincter of Oddi, and helps regulate the passage of bile and pancreatic enzymes into the small intestine, empties into the duodenum of the small intestine. Okay, this again, as I was describing before in one of the previous images, we can see significant portions of what's called the biliary tree, because it kind of looks like the tree, all in green, where we have the gallbladder, the cystic duct, the common hepatic duct, and the common bile duct. The gallbladder stores and concentrates bile. Bile emulsifies fats. That's not the only thing that's found in bile, but as far as digestion is concerned, that's its contribution to digestion. Emulsifying fats means it acts kind of like a phospholipid, where it has one end that likes to interface with a fat, which is non-polar-covalently bonded, and then the other end likes to interact with something polar-covalently bonded, like water. So they will kind of form a circle. It'll surround, a fat molecule or some fat molecules to break up a large mass of fat into small little beads of fat, so to speak. And that helps accelerate in the digestive process of lipids. Cystic duct, hepatic duct, technically it's the common hepatic duct, and then the common bile duct, all for passage of bile. Remember, also in bile are portions of the worn out red blood cells, that conjugated bilirubin. Okay, if we're looking at the undersurface, here we have a liver, and we're looking at the gallbladder here in green, and where we have the green, the blue, and the red meat is the hepatic triad. The green is the common bile duct. The blue is the, or let's take over the red. The red is the hepatic artery, and then the number 2 is the, I just went blank on it. Hold on. Is the vein, that's transporting from the digestive organs. I just went blank on the whole darn name. You can tell when I've been talking a lot during the day because I start to get a little hepatic part of it. I'm so sorry. I don't know why I went blank on that. I think it's because I need some more Starbucks, perhaps. Okay, so we have the liver, over 500 functions. You don't have to know them all, but when you are given an option to match the function, to in a multiple choice environment, the function that generally has the most, or the option that has the most functions associated with it, especially to produce bile, to neutralize or detoxify different drugs and alcohol, produces proteins like albumin, anti-A and anti-B antibodies. That's the liver. Okay, and now we're into the small intestine. That particular region of the small intestine is the duodenum. We have 3 regions to the small intestine: duodenum, the jejunum, and the ileum. The duodenum is the terminal digestion and absorption of nutrients. So we have at the duodenum, it's going to interface with certain products from the liver, like bile. It's going to travel down the biliary tree, and the digestive enzymes produced from pancreas. And when those are allowed to be transported and mixed with the contents of chyme in the duodenum, it completes, it facilitates or provides the enzymes to enzymatically break down certain molecules from um, a macromolecule form to maybe the intermediate molecule, or perhaps from a macromolecule form to a building block in the case of a lipid. So the pancreas, liberates several enzymes to facilitate this process. But as the chyme and the contents of the small intestine move farther down the small intestine, remember it's a very large, narrow diameter tube, is that it will be acted upon even more enzymes produced by certain cells further down in the tube. So where the pancreas essentially merges at the duodenum, we're going to initiate a lot of digestion, of carbohydrates and proteins and lipids, maybe not entirely down to the building blocks, but continues their digestion. As it passes down into the latter regions, of the small intestine, or further down the tube, not completely at the terminal end, is where we have cells that secrete maltase, sucrase, lactase, and dipeptidase. And these are enzymes that will break down carbohydrates and proteins. Oh, and I'm sorry, let me go back. This again, is material really for the next lab, which would be lab 8. Is that also in the duodenum, we release two hormones are produced there. Cysteine kinase, CCK, and secretin. The type of specific physical movement through the small intestine is called segmentation. The specific kind of churning that occurs in the stomach is called gastrochurning. We also have peristalsis because we need everything moving in a forward direction. But in each area, like in the large intestine, we're going to have what's called haustral churning, is that there are some other types of physical movements that are produced by that particular organ. Okay, so here we're looking at the remainder of the small intestine. We looked at the duodenum, which would be, generally will point to a region where the pancreas and the duodenum meet at that sphincter of Oddi. But if we point a little farther down in this long tube system, maybe where 2 is there, or even farther to the left-hand side, I mean, that would be, I think that's a number 13 that I'm trying to point you there. That is the jejunum. Toward the end of this long coiled system, the narrow small intestine merges with the large intestine there at 4. So the region 3, the terminal end or the distal end of the small intestine, is the ileum. And where the ileum meets the large intestine here at 16, we have 15, which is the sphincter, or 4 is pointing to that sphincter, and that is the ileocecal valve. And so the ileocecal valve, like any valve or sphincter, it relaxes and contracts. It allows material from the small intestine to enter into the large intestine, to the cecum, to the left-hand side, and constricts to disallow or discourage the bacteria, that normally resides in the large intestine from backing up and entering into the small intestine, where you have a lot of mucus along this entire system, but you really don't want, that kind of bacteria in the small intestine. It's not designed the same way the large intestine is to inhibit them from, getting across the mucosa and into the body, or at least invading the submucosa and so forth of a small intestine. Now, it has mucus there as a nice physical barrier that we studied, but it's not designed to withstand, what's called opportunistic bacteria to reside in there. So we have two. I don't know what happened to is one, but the two is the jejunum, 3 is the ileum, and 4 is the ileocecal valve. All right, then we have, and I'm not sure what happened to the answer for those, but that is what those are. What happened to the other card that should go with it? I could probably make one and just put it up there. Then we have to turn this up right side up. We looked at this model when we were looking at the lacteals of the lymphatic system. So now we're back here because this is from the small intestine, and we can see layers going across. We can see the mucosa from about midway in that blue Peyer's patch upward. All right, that's your mucosa. And then through the center there, kind of where 2 is located, that would be the submucosa. Then you have the muscular layers, the inner circular, which would be 3, and the outer longitudinal would be 4. And then the connective tissue that would be below 4, because of the areolar connective tissue, that would be part of the serosa. So you have mucosa, submucosa, muscular layer, of which there are two types of muscle, circular and longitudinal. And then you have number 5 pointing to the serosa outside. All right, so here we have the mucosa layer. Those little finger-like extensions are the villi. That's where we saw the lacteals. They help to increase the surface area, slows down the movement of chyme through there, just allow the digestive process to occur, and again allow more absorption of digestive molecules. The submucosa, the muscular layer, 3 was the inner circular, 4 is the outer longitudinal layer. And then 5, which was toward the bottom there, that is the serosa, composed of areolar connective tissue. Okay, so here we're looking at the cecum, which is the little pouch of large intestine, so to speak, inferior to the ileocecal valve. And then further in purple, number 24, that's your appendix. Then we have the regions of the large intestine, the ascending, transverse, ascending at 3, transverse at 4. It's going to travel across, over to the far right-hand side where you're looking, and make a hairpin turn, or what's called the splenic flexure. And then you have the descending colon to the sigmoid colon, and then the rectum at 23. So the large intestine, the function of the large intestine has multiple function, but it mostly stores and forms feces. We have normal bacteria there that are there to produce some vitamins, like some B vitamins, vitamin K, lipid soluble vitamins like I just mentioned, A, D, E, and K can be absorbed there, as well as some other lipid soluble substances. Most water has already been absorbed in the small intestine. Your nutrients, the majority of nutrients, or really virtually all the nutrients, with the exception of some of the vitamins, were absorbed in the small intestine. So the large intestine, pretty much you're done with nutrients, with the exception of some vitamins and a few lipid soluble molecules. Now you're really just preparing to eliminate, to produce a form of feces that you can eliminate. And so we have 1 is the cecum, 2 is the appendix, ascending colon, transverse colon, ascending large intestine, transverse large intestine, that's fine too. Okay, so now we find that go all the way across. Now we have one, we're going to take the descending colon of the large intestine. Two represents the sigmoid colon that sort of takes an S turn and kind of travels kind of not straight back, but back toward your sacrum before it forms the rectum that is more of a vertical portion of the large intestine at 3. Okay. This is a slide of the stomach. Generally don't ask you that. So we're just going to skip over that. If you'd like to look at it, you can. I just don't ask. And then we have, lastly, well, not lastly, it'll be the pancreas. We have the small intestine. You didn't even put a card with it. All right, so we have the small intestine, and we have the mucosa, which is the upper portion. It's mostly darker purple staining, and you can see kind of a line between that material there and that which is below it, which is more of a lighter purple, light pink staining region. You have the mucosa, submucosa, and you can actually see, if this is magnified a little bit more, the glands, you can see those simple cuboidal epithelial ducts that form glands clustered. And then below that, in the purple layers, we have the thickest kind of purple pink region. That is your circular muscularis layer, and then to the periphery, to the outer layer, down toward the lower left-hand side, that would be the longitudinal muscular layer. This is a slide of the liver. That one unit, kind of octagonal, kind of looks like a stop sign. That is one lobule. Next to it is another and another and another lobule. The cells we find in there are hepatocytes. If we can magnify this a little bit, we will see these little capillary-looking structures called sinusoids that are passing blood, down to the hole in the center called the central vein. All right, so here's a little liver lobule. You have the central vein inside. We have the hepatocytes scattered through here. They form these sinusoids. They're kind of like a canaliculi or a capillary between one lobule to another. All right, we have those hepatic triads. All right, hepatic triads. That is composed of one hepatic portal vein, which is the largest vessel you'll see. And then similar size usually is your hepatic artery and your bile duct. The hepatic artery will have a muscular wall, and you'll often see some red blood cells in there. The bile duct will be composed of simple cuboidal epithelial tissue, right? Because like 99% of ducts are composed of simple cuboidal epithelial tissue, and it's devoid of any red blood cells in there because it's not transporting blood, it's transporting bile. It's important kind of recognition and to keep in mind. This is showing you one of these portal triads within the liver. Here we have a hepatic triad. We have an artery here to the about 3 o'clock. You can see like a muscular wall, and you can see red blood cells in the center. To the left, this you can't even see the entire thing. It's so huge. The hepatic portal vein is like the Grand Canyon compared to the arteries and the ducts. Right, so the artery is about there at 3 o'clock. You can see some red blood cells in the center. You can definitely see a muscular wall to it. Over to the left, that is a great big hepatic portal vein. You can only see the right and left kind of wall of part of it. And then about 1 o'clock, you have a duct. That's about that. So here your hepatic triad. He tried to draw you a picture so you knew what you were looking at. Hepatic artery, bile duct, hepatic portal vein. Okay, we're back in the pancreas again. Remember the islets of Langerhans are the pale staining region right in the center. And the pointer here is pointing to these darker staining purple cells that are surrounding it. And those are the acinar cells or acini cells. They function in the digestive system because they're the ones that produce the bile and the different, the bicarbonate, my bad, the bicarbonate and the different enzymes for digestion. Right? So there's your islet of Langerhans with alpha and beta cells. And then the other darker staining purple or pink cells would be the acini cells. Okay, so that's the end of those images. I'm briefly going to open up the notes. I'm going to stop the share and reshare it in the event that it doesn't quite follow us. So hold on. I'm going to download these. Okay. Attempt to reshare that PowerPoint. Okay. Okay. So the digestive lab. Here we talk about the general information. Digestion has two major processes, mechanical digestion and chemical digestion. Mechanical digestion is how we break things down physically, like chewing, or methods of transporting, moving material in through the large intestine, the small intestine, or in the stomach, like gastric journey. And then we have chemical digestion, which is how we use enzymes to break the bonds, of these organic families that we're ingesting. So you can go through this on your own pretty much because it coincides where you can find the answers essentially to the matching and some of the functions and things that I already covered with you. But the mouth, it begins carbohydrate digestion. We have different muscles there, digestive muscles, a temporalis and masseter muscles, aid in digestion. We have salivary glands, the parotid, submandibulars, and the sublinguals. Teeth for mastication, tongue for manipulation of food, taste buds. The pharynx. We have specifically the oropharynx and laryngopharynx. We have the esophagus, the stomach. All right. Yes, the esophageal or cardiac sphincter, the rugae, the pyloric sphincters. Structures, functions, structures, functions. So you can read through those. We enter in the small intestine. We have a duodenum, the jejunum, and the ileum. Major organ for digestion and absorption. Villi, increased surface area. So all those things that I was really referring to are just captured in the notes here. Some instructors ask about the plicae circularis. It's really are the indentations, or let's say they are the folds within the small intestine, kind of like a rugae, but these are around the circumference of the small intestine, and they don't go away when the small intestine is or disappear when the small intestine is stretched. They remain there, and they help to again increase surface area, just like villi do, and help slow down the movement of material. The speed or the transit speed, so to speak, of chyme through the small intestine. Your sphincter of Oddi, large intestine and its functions, the appendix and the cecum, the ascending, transverse, descending colon, sigmoid colon, rectum, ileocecal valve. The liver converts nutrients, produces bile, detoxifies substances, many different functions, deaminates amino acids. We have the hepatic duct, which transports bile from technically a right and left hepatic duct, because you have a right lobe and a left lobe of your liver. They merge to form a common hepatic duct. It merges with the cystic duct to form a common bile duct. Hepatopancreatic ampulla, the sphincter, the hepatic portal vein, hepatic artery, hepatic vein, the central vein. Remember, the hepatic vein is transporting and draining the liver after all the blood from the hepatic artery and the hepatic portal vein has circulated, circulated through the liver. And now all that blood is going to be transported through those sinusoids to the central canal, or the central vein, I should say, of each lobule. And those central veins all merge to form the hepatic vein that then transports blood from the liver to the inferior vena cava. Okay, so these images are really good because they supplement the images that we have on the course page and identify, a few more structures in different ones. So I would go through here. Here's another what we call purple board. We'll find these organs. Basically, as we looked at before. Here we're looking back at the stomach, the different cardiac and pyloric sphincters, the rugae within the stomach. Here's our model of the small intestine, which is at least upright, so it's a little easier to see. Right, we have the finger-like extensions, which are the villi. Within the villi, we have like a D, the lacteal, that transports dietary fats. We have capillaries in there, arteries and veins. We have not that it functions within the system, but we looked at the Peyer's patch, B, the blue lymphoid structure. So that's familiar. The layer that D and A are in, that is considered the mucosa. Where the letter B is, and down to where the muscle layer of number 1 and 2 are, that is your submucosa. You have the 1, which is the inner circular layer of the muscularis layer, and 2 would be the longitudinal layer. Again, your pancreas at B, you have D, which is pointing to the small intestine or the duodenum of the small intestine. A is pointing to the common bile duct as it merges here with the white duct system of the pancreatic duct. C, is the gallbladder. Again, parts of the biliary tree, A, B, C, and D. Slides, stomach, I have here, FYI, that's for your information, but I do not ask you. I will ask you about the small intestine. So these two pictures are very good for the small intestine, and they give you a little more detail. We have the layer A, right, which are those villi sticking, extending into the lumen of the small intestine. The layer is the mucosa. Right? That as a structure would be a villi, and its function would be to increase surface area. Below that layer is B, where we have the submucosal layer. All right, so we have kind of A here over on the left side, and then we have A, B, C, D over on the right side. So A is pointing to the muscularis layer, the inner circular, and then the outer longitudinal. You have intestinal glands that are found here at B, the villi, and it's just pointing to some goblet cells. I generally don't ask the goblet cells, but if you're wondering what those are, goblet cells, just like along the respiratory system, secrete mucus. Then we have the pancreas, lovely picture. B is that islet of Langerhans. And then A is referring to the darker pink purplish cells outside of the islets of Langerhans, the alpha beta cells. They produce digestive enzymes, 7 digestive enzymes, and bicarbonate. Here is a picture of a portal triad. This is a nice one too because it's nicely stained and you can see a little more detail. But here we have an A, the portal vein, B would be the bile duct, and C would be the hepatic artery. It's like a bad example, hard to see the muscular wall, but you can see a little bit of it. Generally, the ducts will collapse on themselves. It will be a single layer of cuboidal cells. There is no muscle in there, so it will not be popped open and maintain any kind of patency. The arteries generally will. Sometimes they'll collapse a little bit, but you'll see a muscular wall. And the veins generally collapse as well, but that was nicely popped open. Here, this is a great picture of It doesn't get the entire lobule of the liver or an entire lobule in the liver, but you can see the fenestrations. There are these sinusoids, that transport blood from the arteries and veins, transporting blood in the hepatic portal vein and hepatic artery at a location like D here. And they travel down those sinusoids toward B, the central vein. And D, you can almost distinguish between, even with this, even far away, right? You can see this great big vein. I don't know if you can see my arrow, but then I'm hovering over this, but the great big vessel, about 8 o'clock is going to be hepatic portal vein. And then the bile duct and the hepatic artery are found at D. And then there's another two, hepatic artery and bile duct, inferior to that hepatic portal vein. So they kind of repeat, around the periphery of these lobules. Teeth, we showed you pictures of teeth that were, in a mandible or in the maxilla, in a mouth, but here they are loose. Right? So we have an A that would be a molar, B would be premolar, C is the canine, and D is the incisor. Function, metafunctions. Here is one of the pictures, again, that we've used in the notes, or that were up on the course page. A incisor, B canine, C molar. There's another. Incisors, canine, premolars, and molars. Cut, shred, grind, and grind. Okay, so when we are using the when we're in the classroom and we have these models, this is just an extensive kind of key when you have it right in front of you for people who want to sit there and go through an entire list and look at the models or the numbers actually on the models. But you don't have to worry about that. And that's it. So some good images in here, some notes that cover a little more detail, some extra in it. I like some of those pictures in here as usual. I use pictures that are from the course page, that we first went through, but you can see in the PowerPoint notes, there's some images of models that are just, very good examples for testing and quiz purposes. All right, well, that's the end of Digestive Lab 1.